Hey 7th graders, I'm going to show you guys your lesson for 2.1 today. Obviously you guys will be doing this in class if you're A or B cohort, so don't try to do this at home unless you're absent. And then obviously DLI, you guys will be doing this at home. So a lot of the directions in this activity are for DLI, again, or those kiddos who are absent. The rest of you guys will be doing this in class, and we'll still be doing the same assignment, but we'll have a little bit different directions as you guys will be using the actual connects to build these proteins, whereas DLI kids, you'll be building them theoretically and drawing what you think they would look like. Okay, so this is a hands-on activity. There's two parts to it. The first part, you guys will be looking at two combinations of gene versions. So obviously we get one from mom and one from dad, and you'll be using the materials to build or draw what you guys think these proteins look like. Okay, so this is kind of what everyone was given if you were in class. So students will be given a bag, either marked A1, A1. So that means that one parent passed down the A1 and the other parent passed down A1 as well. Or a bag A1, A2, where one parent passes down A1, the other parent passes down a different gene, which is A2. Or A2, A2, again, both parents would pass down the same gene version or allele, which is A2. The bag includes all the connects in an envelope. Okay, so the connects look like this. You guys will get to see what they look like here in a second. I'll show you a materials list or a supplies list. And then you also have an envelope inside. And in the envelope, you have a set of instructions. So if you have A1, A1, you guys should have two sets of instructions. One to build an A1 protein and one to build an A1 protein, which, if you put the thought process together, those are both the same. So they should build the same protein because they're the same gene. Okay, if you have A1, A2, you guys again have two sets of instructions. One's for A1, one's for A2. Since those are different genes, you'll probably see different proteins, but we'll find out. Okay, and then finally, if you have A2, A2, same thing. Two sets of instructions, but you're building the same protein because you have the same gene. Okay, read through this little set of directions. It kind of talks to you guys about which, like what the envelope represents and what the connects represent and stuff like that. So you will need this information to answer the questions for activity 1. So if you guys get to activity 1 questions and you have no idea what it's talking about when it says, what does the envelope represent? Well, it's right here. The envelope represents the nucleus. Okay, why? Because it has the instructions to build proteins, and those instructions in our cell are DNA. So we would find the DNA in the nucleus. Okay, so read through the rest of that because it talks to you guys about kind of what the student roles would be. In class, you guys will be working in groups of 3 or 4, depending on how many kids we have. Okay, here's kind of what your bag might have in it, and I put this stuff on here so you can just kind of see what these items are. Again, you may use some of these, you may not use any of them. Well, you'll use some, but you might not use some of them, excuse me. But you guys would basically have these items available to you to build, or in the case of DLI, to draw what your guys' picture would look like. Okay, so you guys can see, obviously, we got these red rods that are longer than the yellow, and the yellow are longer than these blue rods. We have these little gray connectors that kind of connect a little bit more than 90 degrees. These red ones connects at 90 degrees, and these connect straight across from each other, so at about 180 degrees. Okay, you guys can see how the rods and the connectors would connect to each other in this little picture here. Okay, so let's talk about the first part. So you guys will obviously have 3 different combinations of alleles here, or gene versions. I'm not going to have you guys do them all. I'm just going to assign you guys one gene version, and you guys will do the rest of this lab based on that gene version. So if your last name starts with the letters A, B, C, or D, you guys will be doing the gene version A1, A2, where you get an A1 from your mom and an A2 from your dad. Okay, so you guys can see where your instructions are. So read the instructions for A1 on slide 6. So if I click to slide 6, okay, there are my instructions. Okay, you guys can see how to build the A1. And then it says also that you have a second gene or a second protein to build. So your instructions to build the protein for A2 are on slide 7. So you guys would build one protein on A1, one protein on this slide, A2, and then you guys would go down to slide 9 and draw what your A1 protein looks like and your A2 protein looks like. Okay? All right, if you guys have the last names that start with F through M, you are A1, A1. So basically, you guys will only need slide 6 instructions because you're going to build 2 different copies of the A1 protein. Okay, so follow the directions. Imagine what it would look like, again, using kind of this little supply list here. And then you will go to slide 8 and draw your A1 protein here, and your identical, should be identical, A2 protein, or excuse me, A1 protein here. Okay, A2, same thing, except you guys are going to be using slide 7 for your instructions. You're going to build 2 copies of that. And you're going to draw your drawings on slide 10 here. So here's your first A2 protein, here's your second A2 protein, and they should be identical. Okay, when you guys are drawing, you guys will go to your draw feature, and you guys will choose either the pen or the highlighter. It's up to you guys. But know that you guys can change the color. So if I'm in A2, I'm probably using the yellow rods. So I can draw a straight line. It doesn't have to be perfect, you guys. Obviously, I want them to kind of be comparable to size. So my yellow rod, if I look back up here, you might have to switch back and forth to your cursor. You guys can kind of see roughly how big your yellow one is. And you guys, like I said, will only have a certain space to draw them in, so you might not have a rod that looks quite this big, but use your colors to definitely represent what that looks like. And then I would use a red connector, so I'd go and change my color back to red, and it looks like my connector kind of does a little bit of a 90-degree angle here. So maybe my connector looks like that. Again, this doesn't have to be perfect. And then maybe I do a blue rod. So my blue rod would be a little bit smaller. So it might look like that. Okay. Again, your protein won't necessarily look like that. It will have similar shapes. Biggest thing is when you guys are reading your instructions, that you pay attention to the shapes it tells you. So it'll tell you guys, all right, it should look like a wide V shape at this point. Okay, at the end of this A1, it should look like a zigzag shape where you guys go up, down, and up again. Okay, same with A2, you guys have these shapes that are described on here. It's just the matter of which rods and which connectors you guys are using. Okay, so again, if you guys are only doing one of these 3 slides, 8, 9, or 10. So based on what I assigned you guys, I think A2 was for students who are O through W. So basically the lower half of the alphabet. Okay, that's the first one. Then you guys will go and you will answer these questions. So again, anywhere you guys have question marks all over your slide or on your slide, you guys have questions to answer. Here's my purple writing, so you guys will make sure your answer is also in that same purple writing. Again, refer back to slide 3 for the answers to these questions if you guys don't remember what they were from reading through them the first time. Okay, then compare your 2 protein molecules. Are they the same or different? Why? So you guys will have different answers, whether you are A1, 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 A2, or A2, A2. And then finally, question 3 on slide 12. Which claim about the genes' roles in building proteins best fits considering the modeling activity you guys just completed? So you guys just drew those little proteins. So do you guys think that claim 1 is the correct one or claim 2? Again, if you guys aren't sure, think back to this slide right here. Again, your activity 3 slide. Read about what each thing represents and how each member of the group contributed to this process. Okay, and then that should help you guys answer whether you think claim 1 or claim 2 is correct. If you guys think claim 1 is correct, grab a marker and circle it. If you guys think claim 2 is correct, grab a marker and circle it. But either way, I should see you guys having circled an answer there. All right, moving on. So what would happen if we had a mutation in our DNA? Okay, in this activity, you guys are going to be using the same combinations of alleles that you guys used in the first part. So again, if you were A1, A1, you're still A1, A1. Okay, if you were A1, A2, you guys were still on this second part A1, A2, and again, A2, A2, you guys are staying A2, A2. So make sure you guys are filling out the correct information on the correct slides. Okay, so what happens is a mutation has occurred to both of your DNA strands. Okay, so when you guys were reading those instructions to build your first proteins, we now have slightly different instructions. Okay, you will need to remake your 2 proteins following a new mutated instruction. So if you were A1, A1, go to slide 14. Okay, these are your 2 new instructions to make your 2 new proteins. Okay, so if you need to, go back and review what they looked like beforehand, and now think of how you guys would rebuild them following these instructions. Okay, if you guys were A1, A1, go to slide 15. Again, these are your new instructions. Okay, it looks like we have a little bit different mutation than we did in A1, A2. And then if you guys were A2, you guys are going to go to slide 16. Here are your new instructions to make your proteins. You guys will draw on the corresponding side here. So A1, A1, you're going to draw your 2 proteins here. A1, A2, here's your 2 proteins. Here's your A1 and your A2. And then A2, A2, here's your 2 proteins. Again, now they should not look the same if you guys are A1 and A2. They should look different than your first, and they should look different from each other. Okay, same with A1, A2. All right, so let's just take a look at some of these. If you guys have an interruption in the middle, and it tells you guys maybe, all right, attach, like this one here says, lock the end of a 3rd yellow rod to the 2nd red connector. If you don't have a red connector, maybe you changed it to orange, okay, then you guys can't finish making that. If the instructions don't make sense, then you might not be able to finish making the protein. If your instructions still seem okay, it's just maybe a substitution of a rod, but it still tells you guys to hook to the same connector, then you maybe will have a different shape of your protein, or you maybe will just have an incomplete protein, like we weren't able to finish building this, okay, because the last instruction didn't make sense, or whatever the case may be. So you guys will go to slide 20 and you will answer these questions. The change in the instruction caused the protein to either be incomplete, we couldn't finish building the protein. So basically there was something wrong with your instruction set that wouldn't allow you to maybe connect the last rod, or whatever the case. Okay, it may no longer exist. This would be something where you see a change in the beginning of your instructions that doesn't make sense or doesn't allow you to even build your protein. Like you have nothing to build on. It would be impossible to do so, whatever the case. Maybe it just has a different structure. It looks exactly the same, maybe in shape, but the last rod's different, or whatever the case is. Or maybe it has a different shape overall. And then has the same structure. There's really no change to it at all. Okay, so you guys will again look at your 2 proteins, and you guys will kind of tell me which one switched. So you guys can write on there and say, all right, for my A1, it was incomplete, but for my second one, it had a different structure. So maybe you had A1, A1, and you had different structures there. So kind of just tell me which one's which, or which one applies to your guys's different proteins. Okay, finally you guys have, this is the correct configuration for the A2 protein. So predict what the protein may look like if this mutation occurs. So I did not do this mutation on any of your guys' instructions, so I want you guys to think about this. This one just says skip right to step 3, so you don't do step 1, you don't do step 2, and you just start off at step 3. So if you guys had that change, what do you think would happen? Okay, so circle your guys' answer there. Last slide, just to kind of wrap it up. This should start to look familiar to you guys. Do you have any questions? Circle yes or no. If you guys do have a question, please type them. Okay, if it's something that you need immediate help on, please obviously send me a message in Teams and I can help you out, or we can chat if you need. And then finally, once you finish this assignment, how do you feel you did on this assignment? Circle what you guys feel is your description so I could teach this. I own this. I'm still unsure about a few things, or I have no clue what you're talking about. Okay, double check that you are 100% complete, and then you guys can go ahead and turn this in. Again, if you guys have any questions, please let me know. If not, I hope you all are having a great day, and I'll talk to you soon.